I am going to give you a quick introduction about the course. I'd like to start by talking about the exam because that's like our aim here. Now, the examination is 1 hour 30 minutes. This exam can be taken 3 times a year, so it can be taken in Jan, June, or October, November sessions. Now, the examination is out of 80 and is always 8 questions. If you're only taking AS Biology, then this exam is 40% of your overall. So 40% for Unit 1, another 40 for Unit 2, and 20% for the practical unit, Unit 3. Now, if you're taking the entire A-level biology, then Unit 1 will weigh less because you'll be taking 6 units in this case. Nevertheless, I'm just gonna go and start with the course materials. First topic, the first topic in our syllabus is molecules. So we'll talk basically today about the water molecule. We'll talk about properties of water, and we'll talk about carbohydrates. So these are the parts of the syllabus that we'll be doing. Throughout the course, we'll be doing both the theory parts, those are always written here in black. We'll also be doing the practicals. Now, practicals do come in Unit 1, Unit 2, but they're mainly examined in Unit 3. So Unit 3 is like the practical unit of AS Biology. We're going to start by talking about the water molecule. Now, water is, as you know, is essential for life, but that has to do with the structure of the molecule. The water molecule is arranged in a special way. Now, look at the way the oxygen and the hydrogens are arranged. There's always an angle here. This angle is around 104 degrees. The way these atoms are arranged here makes the distribution of electrons unequal. Now look, these are the electrons here. Those electrons are usually quite closer to the oxygen than they are to the hydrogen. Now, you know that electrons are negative. Now, this unequal sharing of electrons makes this oxygen here, makes it slightly negative. Look at this symbol I use for slight charges. It's like small delta sign. This indicates that this side of the water molecule is slightly negative just because those guys, those electrons here, are closer to the oxygen than they are to the hydrogens. This makes the hydrogens on the other side of the molecule, this makes them deficient, like slightly deficient of electrons. They're not like totally lost the electrons. The electrons are still shared here in the middle. There's still a covalent bond here, but they're unequal sharing. And these hydrogens on the other side of the molecule are slightly positive. So each one of them is slightly positive. This turns the water molecule into two sides. There's one side that is slightly negative, and there's one side that's slightly positive. And the entire molecule described this way is described as being a dipole. So di is two, pole means two sides. Basically, there's positive and negative sides of this molecule. Remember, we've described water as being a dipole. Now, this side is slightly negative, this side is slightly positive because of the unequal sharing of electrons. But think about it this way. What if I've had many water molecules right next to each other? This oxygen is slightly negative, but the hydrogen nearby is slightly positive. So what would those two atoms from a distance, what would they do to each other? They will start attracting. When they attract, this attraction is quite weak, much weaker than the covalent bond found within the water molecule, but nonetheless, this weak attraction is quite significant because it helps to attract water molecules to each other. Think about it this way: water always forms drops. Like wherever you splash water, there's always water molecules clumping to each other. This clumping of water molecules to each other is known as the hydrogen bond. So what's a hydrogen bond? It's a weak bond, yet it makes big difference because there's too many of them. Why is it called hydrogen bond? Obviously because it always involves hydrogen. It has to be hydrogen attached to an oxygen. It could also be, as we're going to see later on in syllabus, it could also be hydrogen attached to nitrogen, as we said, oxygen, or even hydrogen attached to fluorine. But we're not really interested in the fluorine part because we don't have fluorine in biology a lot. But think about it this way: whenever hydrogen is attached to a nitrogen and oxygen, the hydrogen in one molecule will attract to an oxygen in a neighboring molecule, and this is known as the hydrogen bond. Hydrogen bonds are quite important for life in many ways. Now, look at this drawing here. This shows you how the water molecules in a drop, they're attached to each other. So look how the the molecules of water forms like a drop, and this drop has the waters attached to each other. Now, remember, hydrogen bonds attach the waters to each other, and this attraction of water molecules to each other is what we call this cohesion. So cohesion is basically water molecules sticking to each other. Why is this so important? This is quite important because every time a water molecule attaches to its neighbor, then these water molecules will pull each other. So that's quite important, especially in blood vessels and in vessels in plants like xylem, because that helps the water move all together from one area of the plant to the other, or from your feet back to your heart. So as you can see now, each water here is hanging to other water molecules and helps the flow of fluids. Could be water, could be blood, because blood is like, there's a lot of water in blood, you know. So that's what helps the flow of materials, or flow of fluid, I should say, from one side of the plant to the other. You could see this quite often. You could see that how, just like take a coin and put a drop of water on it, you see that how water sticks to each other. The molecules of water stick to it, but water molecules also stick to surfaces. They're quite sticky. This sticking of water molecules to surfaces, which helps the flow of fluids, is what we call adhesion. So adhesion is like when water molecules stick to surfaces. In this diagram here, you can see that the water sticking to each other and sticking to the walls of the stem, the xylem here, helps the flow of water against gravity. That's very, very important, because otherwise water and fluids wouldn't be able to flow within living organisms. We'll continue to talk about properties of water molecules. Those are related to hydrogen bonding. Another property is the high latent heat of water. What is latent heat? Latent heat is how much heat it takes to change the state of a liquid from liquid to gas, basically. In the case of water, the latent heat is quite higher than usual. So it takes a lot of heat to, for instance, to turn water into the gas phase. That's not the case of liquids. Think of ethanol, alcohols. Alcohols have much lower boiling point, or it takes less heat to turn alcohol into a gas than the case of water. Why is it the case of water, why does water have much higher latent heat? Why it takes a lot of energy to change the state of water? That's because to change the state of water, you'll need to break those hydrogen bonds. It takes a lot of heat energy to break those hydrogen bonds. For that reason, the latent heat of water is higher. Now, why are we studying this here? It's because living things are mostly made up of water. So if the latent heat of water was lower, then it would have been a trouble for all living things, because we need to keep water in the liquid phase within our bodies. Another property of water that has to do with hydrogen bonding is the specific heat capacity. The specific heat capacity of water is quite higher than usual, higher than other known liquid to us. Again, specific heat capacity, it's how much thermal energy, how much heat energy it takes to raise the temperature of water by 1 degree Celsius. In the case of water, it takes a lot of heat energy to turn the water temperature by 1 degree Celsius for the same reason. So it's because of the hydrogen bond. It's because you need to break those hydrogen bonds that exist between water molecules to raise the temperature of water. One important question in the exam is about the ability of water to dissolve different solutes. Now, a solute is something like salt or sugar. Water is very successful solvent, and that's very important for life, because for us to transport salts and sugars around our bodies, they have to dissolve in water. Now, how does water basically dissolve salts? So what do I have here? I have sodium chloride, that's like table salt. And I'm going to show you how does this dissolve in water. Now, once you drop crystal of salt into water, the first thing that happens is that the water comes in between those ions of the salt. So like the oxygen from water attracts to the sodium because they got opposite charges. Sodium has positive charge, and the oxygen in water has a negative or slight negative charge. Same applies to chloride. Chloride comes and orients towards the hydrogen, and this basically prevents this chloride and sodium, prevents them from getting attracted, because another water will come in between here and will prevent any further attraction. So now you can see that with more water molecules, all of the salt will get dissolved. So if you've been asked like why water is such a very good solvent, it's simply because it forms bonds with the solutes. Here is a brief description of what happens when salts dissolve in water. So first thing that happens is that the water molecules come in between those salt ions. They get attracted, so like hydrogens from water attract to the negative ions, oxygen from water get attracted to the positive ions, and afterwards then we get what is known as hydration shell. So what's a hydration shell? A hydration shell is like a bunch of water molecules surrounding the ions, preventing them from getting attracted to each other, and that's why the ions are very well dissolved in water. We will change the topic. Now we'll talk about carbohydrates or sugars. So we'll start by talking about simple sugars. These are known as monosaccharides and disaccharides. Later on, we'll talk about polysaccharides. These are like complex sugars. So let's start by talking about mono and disaccharides. Before we go further talking about carbohydrates, we need just a little bit of chemistry knowledge. So quite often they ask you to draw molecules. So for instance, if I am to draw an organic or biological molecule like carbon, where there's carbon, carbon always has to make 4 bonds. So carbon is an atom that has to form 4 bonds. Hydrogen, on the other hand, forms just a single bond, while oxygen forms 2 bonds. So these 4 bonds for carbon could be like in any form, like double bond, double bond. Same for oxygen, it could be also just a single double bond. So just try to remember those number of bonds. Try to remember that carbon forms 4 bonds, hydrogen forms 1, oxygen forms 2 bonds. Good thing in biology we don't study many atoms. These are like the main atoms that we deal with. We now talk about the chemical reactions, the essential chemical reactions that we need to learn biology. One chemical reaction is known as condensation, and the other one is known as hydrolysis. Now, condensation reaction is the one that helps us to join molecules. So I have those boxes here. Each one represents like a molecule, and I want to show you how do molecules like sugars join to each other. Usually in carbohydrates or proteins, fats, all of these got oxygen, hydrogen. Let's say I also have oxygen, hydrogen. Now, what happens usually is that when you have OH in each one of those molecules, this helps to form a bond. So let's take the OH here on the left, red molecule, and the H in the other. If these are condensed, these are joined together, this means that I've taken away a water molecule. So water molecule has been taken out from the combination of 2 molecules. Since I took away water molecule, this means what? This means that now I'm allowing those molecules to join. Now look, I'm only left with an oxygen. This oxygen now, remember oxygen makes 2 bonds. This oxygen will now help me to form a bond. So like I'm taking away, that's just for neatness here. Look, the oxygen now joins the molecule on the left to the molecule on the right. So again, just briefly do this again. OH, you take away water out, so OH and H. This could be either way, like you could take the OH from the blue and the H from the red, or the other way around. Once I take the water out, this allows the oxygen to form a bond, and the reaction that we have just made here, we call it condensation, because it always involves the removal of a water molecule. So that's now like a bond. The reaction is condensation. Now, hydrolysis is basically the opposite reaction to condensation. So hydrolysis is when I break a bond. Now, let's go back to our molecules. Here I have a bond. Now, what if I want to break the bond? Let's say it's more like digestion. You've eaten something and you want to break it down so you can make use of those nutrients. How do you do hydrolysis? How would you break a bond by hydrolysis? So look at the way I'm going to do it. It's just going to be opposite to what I've done earlier. I add water here. So instead of removing water, I'm going to add water. When I add water, this means I've broken the bond. So the O here, the oxygen, goes here on the right, and OH is on the left. So now I have added HOH, and this has broken the bond between those 2 molecules. So again, this reaction is what we call hydrolysis. That's the reaction we always do when we break bonds. Anyways, now we're gonna do this once again. While we're learning about carbohydrates, we're gonna do a lot of condensation and hydrolysis. So if you didn't get this, actually, we're gonna repeat this several times as we move on throughout this lesson. We're gonna start talking now about carbohydrates. As I told you earlier, carbohydrates fall into two major categories. There are simple sugars and complex sugars. Now, simple sugars could be classified into monosaccharides and disaccharides. Monosaccharides are just simply made up of one unit, so it's like one molecule. On the other hand, disaccharides, think of sucrose, that's like the one you add to your tea. Disaccharides are made up of two units. So simple sugars are monosaccharides, disaccharides. What do they have in common? What they have in common, those simple sugars, is that they are water soluble. So they're simple, they could drop sugar in water and it dissolves quite easily. But nonetheless, monosaccharides and disaccharides are quite different in some of their properties. So again, simple sugars, monosaccharide, think of glucose, the one you have in grapes. Disaccharides is the one you have in sucrose or lactose, the sugar of the milk. And they are both quite similar because they both dissolve well in water. We're going to now start talking about monosaccharides in more depth. So we're still in simple sugars. We're still talking about monosaccharides and disaccharides, but I'll start with monosaccharides. Now, what is special about monosaccharides? Only monosaccharides have this general formula, which is unique for monosaccharides. So in each monosaccharides, you got carbon, hydrogen, 2, and then O. What does this stand for? Now, every monosaccharide has to have this ratio between its atoms. So for every carbon, you got to have twice as many hydrogens, and for every carbon, you have to have equal number of oxygens. So think of glucose. Glucose is C6H12O6. You can see it's the same ratio, twice as many hydrogens as carbon and oxygen. So this applies for every single monosaccharide. Only monosaccharides got this unique ratio between its atoms. If you look closely, you realize it's more like water molecule here. It's like H2O. So the ratio that we have here is quite similar to the one you have in water, because twice as many hydrogen as oxygen. And that's where the word carbohydrate comes from. It's carbon hydrate because there is this unique ratio of 2 hydrogens for every oxygen. What's special about monosaccharides is that they are the building blocks. So they are the building blocks for disaccharides and polysaccharides. So to make starch, you need glucose. To make sucrose, you also need glucose. So this is another property of monosaccharides. Monosaccharides themselves could be classified into two groups. One is called hexoses. So I'm just going to type it here, hexose. What does a hexose stand for? A hexose is any carbohydrate or any monosaccharide that's made up of 6 carbons. So you have 6 carbons in a hexose. So hexa is 6. Ose is usually the suffix we use for carbohydrates. Good example of a hexose is glucose. So this is a glucose molecule. It's a hexose because of 6 carbons, 12 hydrogens, 6 oxygens. We usually represent monosaccharides and disaccharides. We represent in diagrams like this one here. Luckily, you don't need to learn how to draw those molecules, but you need to be able to recognize where the carbons, and later on you need to learn how to join them in a condensation reaction. So what I need to do here, look, every angle represents a carbon. And if you count those, we usually start counting here at 3 o'clock. So this is where we start the counting. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and the 6th carbon is there on top. So this is a hexose because it got 6 carbons. What about the other category here? The other category is known as pentoses. So pentoses is something we're gonna not deal with today because this is for DNA and RNA. So this is for nucleic acids or genetic materials. So I'm not gonna deal with it here. I just want to show you how do those pentoses look like. I have an example here. I have this pentose. So again, remember, to count the carbons, you start here. This is carbon 1, carbon 2, the angle, carbon 3, carbon 4, and the 5th carbon. So that's basically what are the 2 categories of monosaccharides. Remember their general formula, remember that the building blocks, and remember that they fall in 2 categories: hexoses, that's plural, and pentoses. All right. Previously, I told you that monosaccharides and disaccharides are quite soluble in water. Now, how would you explain this? This is another important question. How would you explain the solubility of monosaccharides and disaccharides? Remember, water is a dipole. Now, when you have a monosaccharide like glucose here, the hydrogen from glucose is slightly positive. Reason, because it's attached to an oxygen. This attracts an oxygen from water, and together those hydrogen and oxygen, what would they do? They would form a hydrogen bond. The formation of hydrogen bonds between carbohydrates, monosaccharides, and disaccharides is how you explain the solubility. So if a molecule in general, doesn't have to be carbohydrate, if a molecule in general forms hydrogen bonds with water, plenty of hydrogen bonds, then this would be a soluble molecule in water. We're now going to talk about disaccharides. Now remember, disaccharides are made up of monosaccharides. So look what I have here. I got some glucoses. So I have one glucose here on the right, one glucose here on the left. Now, when you have 2 glucoses right next to each other, this will invite those OHs to condense. Remember, condensation is a reaction where you take away water from 2 neighboring molecules. So look what I'm going to do. I'm just going to take my eraser. I'm going to erase OH from the first glucose on the right. Just going to erase H from the one on the left. Now this oxygen here will automatically form a bond with the glucose on the right. So this oxygen now is joining those 2 glucoses together and have formed what we call a glycosidic bond. So this is bond that forms between 2 monosaccharides. So now the entire molecule that I have here is what I call a disaccharide. Let's check the formation of the disaccharide again. What I have here is a glucose and another glucose molecule. And as we've seen earlier, each one of those, once they are adjacent, we take away a water molecule, all right? And the water, once it's taken away, the oxygen joins those 2 glucoses. We end up with a disaccharide. Now, we're going to have to give a name to this bond. Look at the name. We call the bond, we call it glycosidic bond, but we have to indicate the position of the bond. So as we've seen earlier, we number the carbon, starts from here, 2, 3, 4, 5, 6. Same we do here, 4, 5, 6. Now, since the bond is between the fourth carbon and the first carbon from these two glucoses, that's why we call it 1,4-glycosidic bond. So 1,4-glycosidic bond is the name of the bond that forms between two adjacent glucoses in a disaccharide like the one I have here. Moving now to polysaccharides. Polysaccharides, we learn about one plant polysaccharide, which is starch. So polysaccharides, we're going to look at starch. And we're also going to look at glycogen. So this is our target for the remaining of this lesson, is to learn about properties and structure of polysaccharides. So let's start with starch. Starch is the polysaccharide that's used to store energy in plants. Where would you find starch? Basically, you find it in many plant cells. It's quite useful to store starch rather than storing glucose or sucrose. One important reason here is because it's insoluble. So being insoluble in water, meaning that it could stay inside the cell and remain there without dissolving in water or escaping out of the cell or even affecting the osmotic pressure of the cell. Because if this was, let's say, glucose all over the cell here, glucose is soluble, then that would attract water into the cell and that would cause more water to enter, and that will affect the pressure and the water content of the cell. So plants rather store starch than storing anything else. As you can see, structure-wise, starch is made up of many glucoses, all attached together. Same bond, glycosidic bond. We have quite very similar molecule to starch. This is the one you found in animals. Basically, in the liver of mammals, you find glycogen. So we're going to look at similarities and differences between starch and glycogen. We're going to start learning first about starch. Now, starch is a polymer of glucose. So the entire molecule is made up of glucose. Starch itself is made up of two types of chains. You have one type of chain is called amylose. Amylose is made up of glucose molecules all attached. As you can see, it has like spiral shape. Nonetheless, all of those glucoses are attached to each other in the same way. So it's like nothing but glucoses attached in a linear form. But due to the presence of hydrogen bonds, once again, the OH and H between glucoses, the molecule of amylose, it forms what we call like a spiral shaped long polysaccharide. All right. But starch is not only amylose, because starch is also another type of molecule called amylopectin. So amylopectin. Where is amylopectin made up of? More or less like amylose chains. They're linear, but every now and then you do get some branches like here, here, here. Now, like, what's the point of having two molecules in starch? So it's like, why starch is a combination of amylose and amylopectin? The reason is each one of those molecules provides one great advantage. So let's talk about amylose first. What's the point of being spiral, linear? The whole point here is being spiral, meaning that it would occupy less space inside the cell. So it's like to coil a molecule rather than being linear would save space inside the cell. Plant cells are quite keen to store as much energy in as little space as possible. So that's one great advantage of amylose. What about amylopectin? What's the point of having the branched molecule in amylopectin? The whole point here is because this molecule got to be digested at some point. So the energy is not going to be stored forever. At some point, the energy has to be taken out from this amylopectin. Being branch is great for the cell because instead of being only able to take glucoses from two sides, here I end up having many positions for glucose to be eaten, or another way, to be taken away and used to release energy. So branches in amylopectin provides accessibility for enzymes. So let's say there was an enzyme here. Amylase is the enzyme to digest starch. So when amylase is to break down amylopectin, you got plenty of positions to be digested, and that provides more energy in less space. So again, starch is a combination of two molecules. Each has its properties. One of them is amylose, which saves space, and the other one is amylopectin, which provides more glucoses to be digested at once. Structure-wise, how does amylose form and how does amylopectin form? So in case of amylose, the chains of glucoses are attached in a linear form. So take a look here. What do I have? I have those glucoses, and as you can see, every two glucoses will attach, form a bond, in the same way we saw in disaccharides. So just take away OH, NH, the oxygen remains here and join. So every two adjacent glucoses will do more or less the same thing. And where we end up with here, we will end up having plenty of this 1,4 glycosidic bonds between those glucoses. So again, amylose, the only bond that you have between the glucoses is the 1,4 glycosidic bond. So that's amylose. That's how amylose is, like linear shaped molecule, which helps to save space because soon it will coil on itself and occupy less volume inside the cell. Remember, starch is two things. So starch is not only amylose, this one. Starch is also amylopectin. How does amylopectin form? In amylopectin, another glucose comes along. So now we're drawing amylopectin. So another glucose comes along. So this is my other glucose here. And this time it attaches itself at the position of the sixth carbon. So where is carbon number 6 here? That is carbon number 6. So 1, 2, 3, 4, 5, 6. And carbon number 1 from this glucose, so the OH here and the OH here, those are going to condense and form a glycosidic bond. How is this glycosidic bond forming? Just going to put it just like this here, right? And I'm just going to put it sideways just to show you how the bond forms. So the OH here will remain with an oxygen right here. And this helps to join these glucoses and form a branch. Now, perhaps this is a bit messy, so I'm going to show you another example. So I have another example here, clearer. So that's my glucoses. Remember, this is amylose here, linear. But the OHs in those glucoses, what will they do? They will attach by hydrogen bonds. And those hydrogen bonds is what makes amylose a spiral-shaped molecule. So that's amylose, the spiral form of starch. What about amylopectin? So that's linear-shaped molecule, but check this one out. This OH here, they're going to attach. How are they going to attach? They're going to attach by means of glycosidic bond. And the glycosidic bond now is going to form a branch. Amylopectin, remember, it's a branch molecule. The name of the bond here, now since this is carbon number 6, this is carbon number 1, so the glycosidic bond is going to be called 1,6 glycosidic bond. So again, amylopectin is branched molecule. You have many glucoses. Check this. All of these glucoses are available all at once. So that's great advantage for the cell to get access to all of these glucoses rich with energy to release energy for the cell. All right. So a quick summary about amylose and amylopectin. So first is amylose. It's coiled or helical. It got only 2 terminals for digestion. The only bond available here is 1,4-glycosidic bond. What's the advantage of amylose? The advantage is that it's coiled, so it doesn't occupy much space inside the cell. What about amylopectin? Amylopectin is the branch form of starch. It got many terminals. It got many glucoses available at once. The bonds are 1,4 and 1,6 glycosidic. So every time you got a branch, this is 1,6 glycosidic bond. Finally, we're going to look at the polysaccharides that is found in animals. Now, this is quite very similar to amylopectin of starch, but sometimes students mix this glycogen with amylopectin. They just say glycogen is amylopectin. No, glycogen is a molecule that looks like amylopectin, but is not amylopectin itself. What is glycogen made up of? It's made up of long chain of polysaccharides, all glucoses. You got the 1,4 and the 1,6 glycosidic bonds. So again, whenever you have a branch, that's the 1,6. As long as you're on the mainstream, then it's the 1,4 glycosidic bond. What's the difference between glycogen and amylopectin? One major difference here is that glycogen is more branched. So like we got plenty more branches in glycogen. That is explained by the fact that animals are more active than plants. Animals move about, so they got to have more access to more glucoses to provide energy for movement. Glycogen is found, as I said, in the liver of animals. It's quite compact, just like starch, branched, insoluble. So all of the good things we've learned about starch being insoluble, branched, and compact are found in glycogen as well.